everyone can join us during this series seminar. We expect a lot of interaction with the speakers, and I am honored to present the first speaker of the series, Professor Liliana Quintanar. Liliana did her undergraduate studies at UNAM from 93 to 98,
Everyone can
98 from which she received an honorable mention and the Gabino Barrera. We expect a lot of from the interaction with the speakers during her undergraduate degree. Honored to present the first. She studied for a year at the University of California, Santa Barbara. Liliana worked under the supervision of did her undergraduate for the work on which constituted her undergraduate thesis under the co-tutelage of Ignacio Camacho at UNAM. In 98, she began her doctoral studies chemistry at the Stanford University with Edward Solomon supervising her work. Her doctoral thesis dealt with multi-copper oxidases essential for iron metabolism. After this, she returned to Mexico to do a postdoctoral stay with Lourdes Massieu at UNAM, where she studied the mechanisms of magnesium neurotoxicity. Since 2005, she has been a researcher at Simvestav in Mexico City. She directs a research group focused on the spectroscopic study of metal-protein interactions relevant to the development of neurodegenerative diseases like Alzheimer's, Parkinson's, and prion diseases, and other diseases as well. Liliana has received several awards and recognitions for her work, among which are the L'Oreal UNESCO Prize for Women in Science in 2007 and Research Prize of the Mexican Academy of Sciences in 2017. She will discuss her latest research, and we are very pleased to have you here, Liliana. Thank you.
Thank you very much, Braulio, and thank you to the whole organizing committee for the invitation to this great space called Latinx Chem. It's really a pleasure for me to be here this morning with you and be able to tell you about some of the research that we have done in the past years at the Department of Chemistry at Simvestav. And for those of you who may not be familiar with what is Simvestav, Simvestav stands for Centro de Investigación y de Estudios Avanzados. We are a federal research center that depends directly from the Ministry of Education, and our main mission is to do research and to offer graduate programs in different areas of science and technology. And we have campuses at different states of the country. One of the main organizers, Gabriel Merino, is in Simvestav Mérida. And I'm at the Department of Chemistry that's in the main and oldest campus in Mexico City. So first of all, I want to start my talk making sure that we're all on the same table regarding the importance of metal ions for life as we know it today in our planet. And it turns out that metal ions like copper, iron, manganese, zinc, that are often called trace elements, are actually very important because they're cofactors of enzymes that catalyze the most important redox reactions that sustain life as we know it in our planet today. And here I'm just showing three reactions that I think are very important. Of course, there are many more, but I'm thinking about oxygen activation and reduction. That is something that happens in the mitochondria of our cells every day. And it happens thanks to cytochrome C oxidase, which is an oxidase that depends on copper and iron. And there's many other oxidase and oxygenases depending on these two redox active metal ions. And of course, all of the aerobic life in the planet wouldn't be possible if it weren't for plants that do photosynthesis, and that's the reverse reaction, water oxidation to oxygen. And they can do that thanks to the fact that they have an oxygen evolving complex that depends on manganese and calcium. Last but not least, plants would not be able to do this if there weren't bacteria in their roots that can catalyze the reduction of nitrogen to ammonia as part of the nitrogen fixation cycle. And this is a really hard reaction to make, and yet these bacteria can do it at room temperature and atmospheric pressure, thanks to the fact that they have nitrogenases that depend on molybdenum and iron. So these reactions are really reactions that for chemists are really, really hard to do in a chemical laboratory. And we have yet a lot to learn from how these metal ions do it with metalloenzymes, and how can we mimic that with small molecules. So these same metal ions that are important for life, essential, copper, iron, zinc, manganese, have also been implicated in protein aggregation processes and degenerative diseases. And here I'm just displaying some of the systems that we have studied in the past years in my group. And I'm talking about the prion protein, that it's involved in all of the prion diseases, and it's a metal copper binding protein. The amyloid beta peptide, that it's involved in Alzheimer's disease, and it's also a metal binding peptide. Or the Parkinson protein, the alpha-synuclein, that it also has different binding sites for metal ions. In pancreas, we have amylin, or IAPP, that it's a peptide that aggregates and forms this kind of amyloid aggregates in type 2 diabetes. And it's a peptide that can bind copper and zinc. And today I'm also going to tell you about crystallines that are in the human lens, and metal ions can bind and cause this aggregation. In all these cases, these proteins are metal binding proteins. We still don't understand what is the physiological role for these proteins having these metal binding sites. However, we know that that interaction can also be deleterious and cause aggregates and cause cell toxicity and degeneration. So because of that, in my group, we ask three main questions. First of all, how do metal ions interact with these proteins? What is the coordination chemistry behind this interaction? Secondly, what is the impact of metal ion binding to the protein in the folding of the protein and the ability of the protein to form these aggregates that are associated to cell degeneration? And last but not least, because we love copper, so we center about copper. Copper is a common factor in all of these metal binding proteins, and it's also a redox active metal ion. So we ask, what are the redox properties of these metal protein complexes? And by answering these three questions in all of these systems, we're hoping to get a better insight of what is the role of the metal ion in protein aggregation, in redox activity, in cell death, in all these diseases, and provide the basis for the design of molecules that will have therapeutical potential for these diseases. So that's really the outlook of what we do. And because of what we do, we actually go to many different conferences on different areas of chemistry. We go to inorganic chemistry conferences to learn about, coordination chemistry. We have also been very close to the protein biochemistry community. We have learned a great deal about protein folding and aggregation. And as Braulio mentioned, I have done some work before on neurochemistry, because that's kind of my second love after just inorganic chemistry and bioinorganic chemistry. So we do a lot of, we do collaborate and talk a lot with the community in neurobiology. And I'm mentioning these because in the past month, Mexican science has had 3 big losses. And it's actually people that we happen to know because we are involved with these 3 different communities. And I'm talking about Rafael Moreno from the School of Chemistry at the National University of Mexico, Alfredo Torres Larios, a really young colleague who was a protein crystallographer and was working at the Institute for Cell Physiology at UNAM, and Ricardo Tapia, also from the Institute for Cell Physiology. And I will not exaggerate if I were to tell you that Ricardo is really the academic father of a large percentage of the neurobiology community in Mexico. So these are 3 big losses to Mexican science, and I would like to dedicate my talk today to these 3 dear colleagues, and we're really gonna miss them greatly. So today I'm gonna tell you about 2 stories. First, we're gonna talk about a wrestling match for copper at the synapse, and this has to do with the prion protein with copper and also Alzheimer's disease. And then we're going to move on to the islands to talk about the crystallines and how metal ions like copper could be causing this aggregation. So I mentioned metal ions are essential for life, and I hope I convinced you of that. And our brain is not the exception. It's the organ that consumes the most oxygen. It consumes 20% of the oxygen that we breathe. So of course, it has all this machinery of metalloenzymes to deal with oxygen, like cytochrome C oxidase. But it also has enzymes that are highly specific for the metabolism of neurotransmitters. And just to set an example, dopamine beta hydroxylase, that's a copper-dependent enzyme. So in fact, some regions of the brain may be more enriched by some metal ions more than others because of this kind of metabolic requirement. So the fact that metal ions are essential for our brain, it also requires to have a very complex machinery of how this metal ion is trafficked from the bloodstream, crossing the blood-brain barrier and getting into the brain and being trafficked there in a safely manner. However, during aging, we know that, or we suspect, because it's actually very little studies, about how metal homeostasis is affected during aging. We get a glimpse of this from people studying metal homeostasis in neurodegenerative diseases. So for example, thanks to the work of people like Rosanna Squiti in Italy, we now know that the total amount of copper in Alzheimer's brain is decreased as compared from a healthy brain. But also we know that the amount of copper, the total copper in blood plasma, it's also increased, and that the speciation of the metal ion is changed. Copper is normally bound to metalloenzymes like ceruloplasmin or bound to proteins like albumin. And we know that in Alzheimer's patients, the amount of copper bound to these proteins decreases, and there's more of the copper that's called labile copper, or not non-protein bound. And we think this speciation, change in speciation, has to do with how much of the copper really gets to cross the blood-brain barrier. So we know that there's these really intricate mechanisms and that they might be failing in aging and in neurodegenerative diseases like Alzheimer's disease. But also our brain, I want to make the point that metal ions are not only important as active sites in enzymes like cytochrome C oxidase, which is the active site I'm showing you here that activates oxygen for reduction. We know that metal ions can also act as signaling agents, and this was first pointed out by Chris Chang at UC Berkeley. And just to set an example of this, if this were a presynaptic neuron, a glutamatergic neuron, this means it's a neuron that will release glutamate and zinc in the synapse. And this glutamate and zinc will activate receptors in the postsynaptic neuron. This is how it happens. This is how this neuron is sending a message to this other neuron. And so these receptors like NMDA receptors can be activated by zinc and glutamate, let calcium entry into the cell. And this calcium entry will in turn start a signaling cascade that will lead into copper getting loaded into vesicles and then copper being released into the synapse. And when copper is released into the synapse, it can in turn modulate the activity of other neuroreceptors here. So I just want you to have this image that metal ions will not only be, in an entactic state, in these active sites of metalloenzymes, but they could also be being trafficked through the cell or being released by neurons in the synapse and be interacting with different proteins and actually acting as signaling agents. And precisely one of the stories I want to tell you about, it actually started the motivation by knowing that a colleague in the neurobiology field, Samponi, who does a lot of electrophysiology studies, he found that he needs to have copper and the prion protein at the synapse for the NMDA receptors to be desensitized. What does it mean? I'll tell you that these NMDA receptors get activated by zinc and glutamate, and then in response, they let calcium enter into the neuron. However, if that channel remains open and a lot of calcium enters, then that will cause neurotoxicity. Thanks to the prion protein and copper, somehow this receptor gets desensitized, so it no longer lets calcium into the neuron, and that is part of a neuroprotective mechanism, so the normal way that these receptors will work. And for that, copper and prion are required. What Zamponi found is that if he exposes the neurons to amyloid beta peptide, which is the peptide associated to Alzheimer's disease, the beta peptide could cause these excessive calcium entry. So he hypothesized that the amyloid beta could be acting as a chelating agent, taking the copper away from the prion protein. So we asked the question if this could be really happening, based on in vitro studies, like in the test tube, could beta amyloid really be competing for copper with the prion protein? And we have done quite a bit of work on the prion protein, and I'm just briefly going to introduce this protein. You probably have heard about the prion protein from prion diseases and mad cow disease, because its isoform, that it's rich on beta sheet structure, called the scrappy protein, has been proposed to be the infectious agent that causes this group of encephalopathies that are transmissible. So the prion protein normally is anchored to the membrane of the neuron, as it's shown here, and it has a structure region that it's rich in alpha helices, and it has 3 alpha helices, a couple of small beta sheets, and then it has in the N-terminal domain this whole region that is unstructured. We call it intrinsically disordered, and in this region there are copper binding sites, as we know. Now, some of the proposed functions for these proteins have to do with synaptic transmission, the neuroprotection, like the mechanism I just showed you with an MDAR, and also it's been proposed to act as copper sensing and even copper transport protein. So to talk more about the binding sites, in this intrinsically disordered region of the protein, there are 6 histidines that serve as anchoring sites for copper. And thanks to the work from Glenn Milhauser group at UCSC, we know a lot about how copper binds to this octa-repeat region. It's called octa-repeat because there's a peptide with 8 residues that repeats itself 4 times. So this region can bind up to 4 metal ions. And then there's a non-octa repeat region that the sequence is different, as you can see here. And there's a histidine 96 and histidine 111. And we have spent quite some years thinking about these metal binding sites and figuring out how copper binds there. I'm not gonna go into the details, but just to let you know that as I said, thanks to the work of Milhauser and other groups, we know really well what's the speciation of copper in the octa-repeat region. And then thanks to some work with spectroscopy and also in collaboration with my colleague Alberto Vela, we have figured out how copper binds at these 2 sites. And we know that the binding, it's pH dependent. We know overall that it depends on the amount of copper that's at the synapse. So the metal protein ratio impacts the speciation. And we know that in some cases like these sites, we can form 2 different species at physiological pH. So that's what we know about the prion protein. And now we also know that the prion protein can be cleaved right between the histidine 96 and 111 sites, leaving a prion protein anchored to the membrane with a histidine 111 that has a very different speciation because of this free amino group, and releasing a peptide, a long peptide that can also bind copper into the synapse. This cleavage is considered to be neuroprotective. And actually, about half of the prion protein in our brain is cleaved this way. So it's kind of important to understand how the copper coordination chemistry gets impacted by these kinds of cleavages. Now, what do we know of the amyloid peptide? We know that thanks to the work of Peter Fowler, Crystal Huriao, and other groups, and also our group, we now know that copper binds to the N-terminal region that at physiological pH, it can form these two coordination modes. And because of the coordination properties of these two complexes, the redox potentials are widely different. Only one of these species can actually activate oxygen and cause formation of reactive oxygen species. So we already know how these two proteins bind copper. And the question that my student, Janaí Posadas, and you can see her poster at INORC 73, her PhD work basically focused on figuring out if the amyloid beta peptide can indeed compete for copper with the prion protein. And what she did was synthesize peptides that can very well model these binding sites because it's an unstructured region of the protein, and confront them with copper at different conditions to favor the formation of the main different species, the low copper occupancy, the high copper occupancy modes in the octa-repeat region, also the non-octa-repeat sites, and the alpha-cleaved site. And she asked the question, if I form these complexes in the test tube and then titrate the amyloid beta peptide, can this amyloid beta peptide indeed take away the copper from the protein or not? And I'm not going to go into the details because we have very little time, but just to tell you, she did a lot of electron paramagnetic resonance, EPR studies, and also circular dichroism studies to figure out, to answer this question, if the amyloid beta could take away the metal ion. So what she found is that in some cases, it cannot take out the metal ion, so we remain with the same signal as the signal of copper bound to prion. And in some cases, at the end of the titration, she ends up with a signal that is identical to copper bound to the amyloid beta peptide, saying the amyloid beta peptide really can take away the copper there. However, at intermediate points of the titration, she could see evidence of a ternary complex being formed. So at some point in the titration, we can see species like this one, copper bound to the histidine of the prion protein, but also bound to the amyloid beta peptide, so ternary complexes. In the non-octa repeat sites, the story was much easier. In both cases, the amyloid beta peptide wins, so it can really take away the copper from these sites. And in the alpha-cleaved prion protein, because of the nature of this peptide, we actually end up with ternary complexes. So things like this, where the copper is bound to the histidine and the amino group of the prion, and also the amino group and the histidine from the amyloid beta peptide. So the conclusion here is, depending on how much copper we have there, or what is the copper protein ratio, and what are the species that we're favoring, amyloid beta peptide can either take away the copper or form ternary complexes, and only one of the sites really can resist the competition with the amyloid beta peptide. So this, of course, has implications to how the copper prion NMDA interaction could happen, because only those sites where amyloid beta peptide can take away the copper or form ternary complexes, I have it here, so you can either take away the copper or form ternary complexes with the full protein or the alpha-cleaved protein. In all these three cases, the amyloid beta peptide will cause neurotoxicity, and this will be a new mechanism by which it can exert this neurotoxicity. But also it has implications on chelating therapy for Alzheimer's disease. A lot of groups have devoted many years on designing chelating agents for copper to treat Alzheimer's disease. However, we need to be very careful. The motivation is that copper bound to A beta will cause formation of neurotoxic oligomers and aggregates. However, if we are also ripping away the copper from the prion protein, we will be also interfering with these neuroprotective mechanisms. And that's probably why none of the chelating agents that have gone into clinical trials for Alzheimer's disease have made it past, clinical phase 2. Also, I want to just make you aware that the prion protein, because of this intrinsically disordered region that it has, it can interact with many different other proteins that are involved in other aspects of neurobiology, like neuroitogenesis, plasticity, cell migration. And in many of these interactions, we still don't know what is the role of copper and of the metal ion in modulating those interactions. So we think there's a whole field of prion biology, prion neurobiology that needs to be explored from the perspective of the bioinorganic chemistry of copper and the prion protein. So for more details on this work, I invite you to visit Janai's poster number 73 in the inorganic chemistry category. But now I'm out of time, so I'm going to move on to the human lens and the crystallines. So the human lens, it's really a very unique place in our body. It has to be transparent so that light can get through and hit the retina. So you can see this presentation text to the fact that your human lens is transparent. And it's transparent because the cells that conform it are organized like an onion kind of shape like these, but they also were differentiated during the embryonic stage. So when you were in your mother's womb, these cells started to differentiate and to elongate. They stopped having a nuclei or a mitochondria, and they have no organelles. They cannot produce new proteins, and they cannot degrade damaged proteins. So the proteins that have been recruited to the human lens are highly stable and soluble, and this is a requirement for the transparency of the lens. So the crystallines you have right now in your human lens, that's all you've got for the rest of your life. Your lens cannot produce new proteins. Who are these proteins? So we have the alpha crystallines that are multimeric and they act as chaperones. And we have the beta gamma crystallines. The beta crystallines are dimeric and the gamma crystallines are the monomeric members of the family. I'm going to tell you a story about gamma D, gamma C, and gamma S, which are the monomeric members of this crystalline family. And they're actually among the more abundant in the core of the lens. So you could be asking, how come if these proteins are so stable, how come they end up forming aggregates and causing cataract disease? Well, throughout our lifetime, they're exposed to oxidative damage, to UV radiation, and all these things can cause the formation of partially folded intermediates that will be prone to aggregation. Just to put an example, UV radiation could be opening the indole ring of tryptophan to form kynurenine. This is a more polar moiety, and because the tryptophans are in a hydrophobic pocket of the protein, it will cause partial unfolding. This is the kind of thing that could be happening if you go to Cancun or you go to a nice beach and you are, sipping your margarita or your beer, and if you're under the sun and you're not wearing sunglasses, this is the kind of damage that could be happening to your crystallines. However, you don't go to Cancun and come back with cataract disease. That's because, lucky for you, while you're sipping your margarita at the beach and not wearing sunglasses, you have these alpha crystalline that act as chaperones. They can recognize these partially folded intermediates and form soluble complexes, but they cannot correct the folding. They can just only keep them out of trouble. Eventually, if you keep going to the beach and being under the sun without wearing sunglasses, it doesn't matter how many margaritas you drink, you won't have enough alpha crystallines to prevent this aggregation. So eventually, at a late age, you will develop cataract disease. You must be wondering, what do metal ions have to do with this? So that's exactly the question that I asked. I asked Jonathan King from MIT when I met him about a decade or more ago. I heard his talk about crystallines and I asked him, what about metal ions? Are there metal ions in the lens? I mean, it's low metabolism, but you still need to control, some redox, and you need to have catalases and peroxidases and so on. So he said, we don't know, but we could find out. He had been studying these proteins for decades. He knows them really well. He actually retired a couple of years back. But he really did great contributions to the understanding the folding of these proteins. So we looked up in the literature and we found that occupational metals exposure can lead to early onset of cataract disease. And we also found from a few old reports that a cataract lens may have up to 10 times more copper than a healthy lens and up to 2 times more zinc than a healthy lens. So we asked the question if metal ions could be one more factor contributing to this oxidative damage and cataract disease. And What we did is, well, I went to his lab for a sabbatical. We took this protein, human gamma D crystalline, and we confronted it with different metal ions. And we found that copper and zinc can induce the non-amyloid aggregation of this protein. And of course, the more metal ion we add, the more aggregation we see. These are turbidity assays. The nature of the aggregates are non-amyloid in nature. And as you can see, the effect of copper is much more drastic than zinc. We have some stories on zinc and other metal ions, but I'm gonna tell you today about copper. We later found that all three gamma crystallines, gamma D, gamma S, and gamma C, they have very similar fold. They are rich in beta sheet structure. All three are susceptible to copper-induced non-amyloid aggregation. So we ask about the mechanism, and this is the early work done by circular dichroism was done by Antonio Dominguez, a former student in my group, and an undergrad, Eduardo Martinez. So they did titrations of these proteins by circular dichroism and added the metal ion. These proteins are rich in beta sheet folding, so they have this negative signal here at 218 nanometers. And as we titrate the metal ion, we can see a decrease in the signal, suggesting that the beta sheet fold was being lost. Then if we do thermal denaturation experiments, we could see these curves, the transition temperature, the TM, being decreased to lower temperatures as we add more copper. So basically, this suggests that the metal ions are leading to loss of folding and also loss of terminal stability, and probably yielding the formation of partially folded intermediates that, of course, will be prone to aggregation. Then, thanks to a collaboration with Professor Miguel Costas at the School of Chemistry at the National University of Mexico and his student Oscar Rodriguez, we could do differential scanning calorimetry studies on these three proteins. And as you can see, we confirm that the temperature of the thermal denaturation decreases, goes to lower temperatures as we add copper. Now, this process is non-reversible. When we decrease the temperature, it's not like you're gonna get your protein folded back. No, the unfolded protein can form aggregates. So we cannot extract any thermodynamic parameters from this process, but we can get a sense of the kinetic stability of these proteins and calculate a half-life time for these proteins. This is in the order of years for the wild-type protein, but it goes down 2 to 7 orders of magnitude, depending on the protein, when we add the metal ion. So really, copper ions are significantly reducing the kinetic stability of these proteins. We can also construct the separate domains, the N-terminal domain and the C-terminal domain, and in both cases, turbidity assays indicate that copper can induce the aggregation. But on the circular dichroism experiment where we see folding, only the N-terminal domain is susceptible to unfolding caused by the metal ion. The C-terminal domain remains intact. So what we think is going on is that when we add copper, the N-terminal domain gets unfolded, and we are left with an intermediate where we only have the C-terminal domain folded. Of course, if you increase the temperature, you end up with a fully unfolded protein. So this is what we're seeing here. Now, if we compare the trace of the protein saturated with copper ions to the trace of just the C-terminal domain, the thermal denaturation of the C-terminal domain, we can see that they behave very similar. So this confirms this hypothesis that copper is really interacting somewhere in the N-terminal domain and causing unfolding of that part, that domain of the protein. We also asked if this mechanism of aggregation could have some component of metal bridging. And so Giovanni Palomino, who was a postdoc in my group, he did these turbidity assays where in the middle of the aggregation induced by the metal ion, he will add EDTA. And as you can see, in all cases, he can revert partially the turbidity, so we can reduce the size of the aggregates. In some cases, completely, in some cases partially, depending how much EDTA you add. But in some cases, no matter how much EDTA you add, you end up with still another process causing the growth of the size of these aggregates. So this means that metal bridging is contributing to the formation of these large light scattering aggregates, but it's not the only mechanism. There are other things going on, like we already saw, partial unfolding of the protein that causes formation of the main swapped aggregates and species. So you must be wondering, how is the metal ion interacting with the protein? And this is kind of a hard question to answer, because we need the protein to be highly concentrated for some spectroscopic studies to be able to do. But then at the same time, the metal ion is causing aggregation. However, Giovanni managed to get this beautiful EPR data. I mean, with EPR, even if it's in the aggregate state, we can still see the metal ion. So he found that in all three proteins, we could discern at least 2 different copper-2 species bound to the protein. But something interesting came up, and that's the spin quantitation. So copper-2 is a 3D9 configuration, so it has 1 per electron, and we can actually quantitate the EPR signal and see how much spin we have there. And he could only see 30 to 50% of the copper-2 ions that he added, suggesting that, where did the rest of the copper-2 went? Well, we thought maybe it's getting reduced to copper-1, forming, configuration of 3D10. So then this will no longer be paramagnetic, and we will no longer contribute to the spectrum. Also in the gamma D spectrum, he could see a very stable free radical here, and I'll talk more about that. So these 2 things suggest there's some redox chemistry going on. And thanks to Dr. Niels Schutt, who's a postdoctoral fellow in my group with a DFG fellowship from Germany. And he has a lot of experience on X-ray absorption spectroscopy techniques. So we could also, in collaboration with Thomas Kroll at Stanford, we could collect data on these same samples, the same samples I show you in the EPR, where we see a decreased spin quantitation. And we could confirm the presence of this very sharp feature in the pre-etch, that it's ascribed to a 1s to 4p transition for copper-1. So the D shell is completely full. So then we extract an electron from 1s and goes to the 4p orbital. And we now know from the work of Solomon and Kit Hudson at Stanford that these very sharp features in the pre-etch for copper-1 is ascribed to having a bicoordinate type of coordination. So it yields a very intense electric dipole allowed transition. So in all cases, we can see copper-1 species in the proteins. And also, very briefly, to show you the EPR, the signal of the free radical in gamma D, we can extract the parameters and actually figure out which is the tyrosine that has the right angles, the right orientation to yield these EPR parameters. And we know it's tyrosine 98. So basically the summary is we have this protein that binds copper-2. Copper-2 is causing aggregation, but there's also this redox process. We have some idea of what will be these binding sites in the N-terminal domain, this histidine and this cysteine. But in the C-terminal domain, we have this motif with 2 cysteines that is really reminiscent of copper chaperones, intracellular copper chaperones. And this is happening under aerobic conditions. So the protein is causing this reduction to copper-1. And there's some electron transfer interesting here to look at to the tyrosine, and also asking the question of why this tyrosine-based radical will be so stable. I'm going to skip this. And just to close this, it's just really striking to think about crystallines, these human lens proteins, as transition metal ion binding proteins, and having motifs that are similar to other metal binding proteins. So this motif is similar to zinc binding fingers. This motif, as I said, is similar to ATOX1, which is a copper chaperone that traffics copper intracellularly. So with this, I will end. And the main message is copper ions can induce this non-amyloid aggregation of gamma crystallines. And hopefully I convince you that they do this through different mechanisms. There's some redox chemistry and free radical involved. I didn't show you the data, but we also know that disulfide-rich species are formed. partial unfolding of the protein, and metal bridging. And all these mechanisms lead to the formation of these aggregates. And our next step is to figure out what are the site-specific interactions that turn on each of these mechanisms of aggregation. And hopefully I convince you that metal ions will have a potential role in cataract disease. Just to mention, we have been motivated by this project and also have conformed a really nice multidisciplinary team to study the metallomics and proteomics of cataract lenses. And this is in collaboration with Francisca Dominguez at the Instituto Nacional de Rehabilitación, with Juan Pablo Reyes at INMEGEN, who does the proteomics, and with my colleague Fanny Smith-Sirles at SimbaStaff, who does the metallomics. So hopefully soon I'll be able to tell you about these exciting results on correlating metallomics and proteomics of the cataract lens to clinical, the clinical history of the patient. And of course, we are also doing some theoretical chemistry work on the crystallines in collaboration with Laura Dominguez at UNAM and Alberto Vela at SimbaStaff. And with this, I will end. I hope I convince you that it's worthwhile asking ourselves these three very simple questions in all these systems, and that ultimately we could pave the road to really design molecules with therapeutical potential for these diseases, taking into account the metal ion component or the role of the metal ions in these diseases. And I try to do the acknowledgements as I was showing you the data. But here's the last picture of my group. I've been very, very fortunate to have really great graduate students, undergrads, and postdocs throughout these years. And also a great set of collaborators. I'm not listing them all here, but just the ones that contributed to some of the stories I told you today. And thanks to the funding, and thanks to you for your attention today.
Thank you, Liliana, for such a wonderful presentation. And of course,
I think I was on time, right?
Yes, you were. We have plenty of time for questions, so I will invite the audience to ask the questions directly. If you want to raise your hand or take the opportunity.
We have one on the chat.
Yes, so I'm gonna allow myself to read it out loud. So it says, from Maricruz Rangel, is there any known copper-bound compound that does the function of prion proteins to desensitize NMDA receptors?
That's a very good question. I don't think we know of it. I think what we know is that there must be some interaction of the prion protein with NMDA receptor. We don't know if copper is involved there. Of course, we think there is, but the people who have done these experiments in the past were not really thinking about the metal ion precisely. So we think it's site-specific with the prion protein, but I don't know of any work where they had tried putting other copper complexes and figure it out. There's also some interesting nitric oxide chemistry that we think is going on, and we know that the cysteines get nitrosylated. So it'll be interesting to take some, synthetic copper complexes and figure out if that could have an activity. Yeah.
Okay, thank you. So we have time for a couple more questions from the audience. If you want me to, I will read it out loud, or if somebody wanna take the opportunity. So Liliana, I was wondering. I'm gonna jump in. I was wondering. So it's always how do you, from the organic point of view, organic synthesis point of view, how do you introduce the copper ion? So how's the experimental setup where you can introduce these ions in these very delicate systems?
Yeah, so basically what we normally do is we have our peptide or our protein, and we just add, like copper sulfate or copper chloride, most likely it's copper sulfate most of the times, and the copper ion just binds because you have anchoring sites like the imidazole ring of histidines or a free amino group. So those are really the anchoring sites. I didn't talk much about the coordination chemistry, but once the copper ion is anchored at the imidazole ring or the amino group or both, it can induce deprotonation of the backbone amides, and then those come into place, and then you end up with a very stable complex. The binding affinities of some of these sites go from nanomolar to micromolar in the order, in that order for KDs. So yeah, it's not a problem. It's just the metal ion itself will go in there. Yeah.
Excellent. So we have a couple questions from YouTube, the YouTube audience. So Professor Miguel Mendez says, when you mentioned the glutamate and zinc are released close to the synaptic region, is there any chance they're coordinating each other? So really they are forming a complex?
Yeah, that's an interesting question. So zinc and glutamate are loaded into the synaptic vesicles even before they're being released into the synapse. So they are already in the vesicle. And knowing zinc and knowing it likes carboxylates, yes, as a coordination chemist, one will think it's forming a complex. Yeah, I don't think people in the neurobiology field will normally think of it as a complex. It's just that, two things that are being released at the synapse. But yeah, it could very well be that. And once it's released in the synapse, of course, there's other proteins and other, peptides there that could also take over. Yeah.
Excellent. Thank you. Another question from Angie Forero Giron. Says, thank you so much for the excellent presentation. Are you working on the hydroxylation mechanism of DBM?
No, I'm not. There are groups working on it, like Ed Solomon, of course, he's copper enzyme guy, and D beta H is one of the enzymes that they work on. We don't work in any of these enzymes really, but it's interesting to think about regions of the brain that are dopamine rich and that require copper for this activity. Yeah.
Okay, thank you. So one last question from Professor Jorge Tiburcio from Semestav. He says, from your results, after adding EDTA, it seems that copper may act as the starting seed for aggregation. Is this true? It will be possible to prove it.
Yeah, it looks like that because you saw, the drop in turbidity assay and then like increased again. Maybe I can quickly show. But it's not really because, so if you can see it here, perhaps, Jorge, is that what you were thinking, that it's seeding? This trace comes from here. We add the copper and then that immediately causes aggregation. You don't even need to measure it with turbidity. You can see the solution getting turbid. And then when you add EDTA, it becomes clear. And soon after, without doing anything, just following it in the plate reader, you can see an increase in the turbidity again. So what we think is going on is that these big aggregates that are scattering light, some chunks of it are being held together by metal bridging. So when EDTA, we added like 100 equivalents, like we exaggerated. It's enough EDTA there to chelate all the copper there, and it's such high concentration, it will just really take it away. So you are disrupting that and you form smaller aggregates. But if those are smaller aggregates still have hydrophobic regions exposed, then the domain swapping mechanism that I showed you at the beginning will continue to happen even when you have disaggregated the metal bridging component. I don't know if it makes sense. I think Jorge is here in the audience.
Okay, I think he will reach out to to ask a follow-up question. I want to thank you, and we all from the Latinx SCAM community, we thank you, Liliana, for such a wonderful presentation. And I hope there are more questions, and they can reach out in this email address and the website of yours. Thank you very much.
And Twitter, because we're a Twitter conference, right?
Yes, of course, Twitter.
Thank you. Thank you. It was
Thank you.
really great to be with you today.
Thank you very much. I'm gonna stay here because now we have in a few minutes we have the next speaker, which will be Diego Solís. So if you're interested, please remain here, and I will see you in I think on 5 minutes, less than 5. Thank you very much. for being here at the second conference of this series and the Latinx Chem 2021 event. On Twitter, yesterday we had these poster presentations, and today we have earlier we had a presentation from Professor Liliana Quintanar, and now is the turn of Professor Diego Solís. I'm gonna allow to introduce him a little bit. So Diego graduated with honors with a bachelor degree in chemistry in 2008 from the School of Chemistry at UNAM. Later, in 2012, he received a PhD degree in chemical sciences from his alma mater, also UNAM, under the supervision of Professor Wojtek Jancic. Later, he did a postdoctoral stay at Stanford University with Hamemala Karunadasa, working with inorganic compounds. Now, his lines of research are focused on the perovskite solar cells, hybrid materials, and their applications in photovoltaics, for example, LEDs or solar cells, lead-free perovskites for application in solar cells, and also fluorescent nanoparticles and photocatalysis. But most importantly, this year, Diego was selected as part of CNN News Talented 12, which is a distinction that is given to young scientists whose research work is considered to change the global perception and will have a global impact within the following years. So it's really a pleasure to have you here. And more importantly, I consider him a friend. He's a very nice mentor, supervisor, and very smart guy to work with. Thank you very much, Diego, for giving us this presentation. Go ahead.
Thanks a lot, Braulio, and thanks a lot to the organizing committee for giving me the opportunity to share some of the science that we do. So in a little bit, I'm going to tell you a little bit about perovskites. But before doing that, I would like to tell you a little bit of my story and tell you why I believe Latinx Chem is such an important project and such an important event. As Braulio briefly mentioned, my academic journey started at UNAM and finished at UNAM. I did my bachelor's, my PhD, and now I'm a professor at UNAM here at the Instituto de Materiales. But also during my academic journey, I was fortunate enough to go to different places around the world and to further my education. And when going abroad, I often receive questions or comments regarding the science in Mexico. For example, I would get questions such as, can you do science in Mexico? Can you get a PhD in Mexico? Do you have instruments to do science? And at first it took me by surprise, those comments. I didn't know what to make of them. And later on, I took them personal and a little bit annoying. So I made that one of my personal goals, to show that Latin Americans and people doing science in Latin America can do a great job, can do great science, and can really contribute to the entire community, to the entire world. And this is where Latinx Chem plays such an important role, because Latinx Chem fights that bias or fights that lack of information about Latin American science and Latin American chemistry by showcasing all of the amazing science that Latin American scientists or people in Latin America are doing. And I think that is tremendously powerful. And for that, I congratulate the organizing committee. I congratulate all of you that are here listening to me and to the other seminar. And I congratulate everyone that has put a poster. We've seen a lot of really great science going on. And this is a great showcase of the science that we can do. And I really think that this is a really important step and a really big contribution to show the Latin American chemistry. So congratulations to everyone, and thank you for being here. I think it's really a great event, and I'm sure it will continue to grow and continue to help disseminate this message. Okay, so having said that, as I said, I'm going to tell you a little bit about perovskite solar cells and perovskites in general. And by now, many of you have probably already heard in the news, in the videos, or in social media, something about perovskite solar cells. There's been quite a few articles or posts. And more recently, there has even been some like more traditional news outlets such as NBC make some noise about perovskite solar cells. You might have even heard or seen a graph such as this, in which they show that perovskite solar cells have gone from less than 4% to well over 25% in less than a decade. And also that this is extremely fast as compared to other technologies such as crystalline silicon. So there's definitely a lot of excitement in the community, and there's a lot of potential application, not only on solar cells, but there's a lot of potential in these materials. But I want to tell you first what's a perovskite and things that are not as discussed as I think they should be. So first, I would like to get everyone in the same page and talk about what is a perovskite. So there are two uses of the word perovskite. The first is when we're referring to the mineral calcium titanate. Calcium titanate was first discovered in the 19th century by Gustav Rose. And it's this mineral with this particular formula and this very particular structure, which is formed by these octahedra that are sharing their vertices and forming this three-dimensional structure where there is these cavities that host another cation here. In this case, calcium. Since this was the first structure like this one reported or shown, from then on, like every material that has a structure that is similar to calcium titanate, we call a perovskite. And these days, the term perovskite has been used quite liberally, and many things are called a perovskite. So that might be a topic of discussion for another day, but remember that it's just similar. It doesn't have to be exactly the same, although most perovskites, and by the most traditional definition, it should also have this sort of formula. As I showed you before, like it's not only calcium titanate, but you can think about replacing all of the atoms in that formula. Here, for example, when I replace oxygen by nitrogen or sulfur, the charges changes, and now I have completely different elements in the other positions. So by changing the elements, you can incorporate pretty much any element in the periodic table in a perovskite type structure. And with that, changing that composition results all kind of different properties and all kind of different applications. Just to give you a clue, perovskites can have such a wide range of properties or applications as conductors, semiconductors, insulators, and even semiconductors, of course, depending on the specific composition or a specific formula of that particular perovskite. They can have ferroelectric, piezoelectric, thermoelectric. They can be used as ionic conductors, membranes, quantum materials, and a long list of etc. So they can be used in several different things. One particular family of perovskites is what we call the halide perovskites. Halide perovskites are these where we substitute X, the X position, with an halide, such as chloride, bromide, or iodide. And in particular, this family with lead perovskite has shown to have really interesting properties in the photovoltaic world, as we'll see in a little bit. And another variant of that is when we replace these traditional inorganic cation such as cesium for an organic cation such as methylammonium. So now we have what we call a hybrid perovskite because it comes from meshing the organic and inorganic parts into a single material. So this is a quick introduction to perovskites. And this particular hybrid perovskite, methylammonium lead iodide, that I show here, or similar materials, are the ones that are mostly used for solar cells. And they have a lot of great properties that you might have heard of. For example, they can be made out of cheap precursors. They have high conversion efficiency, as I already showed you. They have high absorption coefficients, which allows you to build really thin and light solar cells. They have PLQEs, which is the photoluminescent quantum yield near unity, which is really good and could be helpful for not only solar cells, but other applications. They have benign defects, which is also really important because it helps you build them from cheap materials. They have good electromagnetic transport properties. Their properties can be tuned, and they have very low exciton binding energy. So these are all great properties. But what most people won't discuss or are not really telling you enough is that they also have some cons. Okay, so first, of course, is the concern about lead toxicity. As you probably know, lead is a toxic element. And there's some concern that deploying lots of perovskite solar cells may end up leaving some lead in wastewater or eventually in food. I mean, it's certainly a concern. Then there's some issues with moisture and light instability, which is certainly not ideal. There's still some issues with scalability. So in short, we can do great cells when they're really small, but if we can do it in bigger areas or in bigger scales, it becomes really challenging to maintain that efficiency that I told you about. And then last, we don't know much about the lifetime of these materials. How much will they last? Having been around only for a decade mostly, it's hard to know how will they respond in a working environment for over 10 years, which is what you ideally would like them to last. Okay, so here is a list, and of course, we look at this, and we, in our group, we try to do two things. To take these pros or these amazing properties and try to use it for different applications or different problems. Or the other one is trying to help address some of the cons of these materials. And we do so by a synthetic approach. We're a synthetic group in our core. So we try to develop new materials for these kind of problems. So to give you an idea, here's a few projects that we have in our group. We work with some luminescent nanoparticles, lead-free perovskites, hybrid 2D perovskites. We try to do some wide LEDs, Cara materials. We do some passivation schemes, and so on and so forth. But today I would like to tell you a little bit about the story about how can we address some of the toxicity and stability issues of the perovskite, and this is how we develop new materials trying to address this. Okay, before I start, this is a work led by these three students, Dr. Brenda Vargas, Maestro Raul Torres, and chemist Diana Castillo. They were the ones that developed most of this work. Of course, we had some wonderful collaboration, but these were the main students working in this, so I wanted to acknowledge their work. And as I said, the motivation was mostly to address the lead toxicity of the materials and see if we can find a suitable replacement of lead in these materials. So what are the alternatives to lead? The first chemical thought would be to go just above or below the periodic table to elements, in this case like germanium or tin. But unfortunately, those are even more sensitive to oxygen. So although they work at some degree, it might be even harder. It's definitely harder to work with these than with lead. So there might not be the idea of replacing to lead. Then you could think of many other 2+ elements that are in the periodic table. But you can also quickly start to eliminate some of them. For example, you can think about of these for being even like way more toxic than lead, or these ones for being way more expensive. And there's a lot of reason why all these others don't work. Copper, it might be an exception, could be really interesting because it's abundant, it's not toxic, and people have indeed tried this. However, the results are not really promising, and the efficiency in solar cells have been lacking like quite a bit. So this was the original report, which reported like a 0.02% efficiency, which is not very good. And there's have been some improvement, but they're still not past the 1% mark. So this doesn't seem like a really promising alternative, and we can discuss why in maybe another time. So what it started like, there's a lot of options, ended up with not that many options. So in come the term double perovskite. And double perovskites are actually really simple to understand. If you get this complicated math formula, which says 2 + 2, it's equals to 1 + 3. I think you can get double perovskites because, so what we do is we replace two metal 2 cations with one which has a +1 charge and another one has a +3 charge. And in this way, we can replace metals that only have this stable oxidation state for another set of metals. And now we can also do permutation of those metals. So how does it look? This is an example. This is not our work, but this is how it can be seen, right? So in here you're replacing a 2+ metal for silver and bismuth, and create an alternative perovskite lattice which has one octaedra of each other. And so this can have really interesting implications. So now you can think about the double perovskite chemical space. Now you can add metals that are only stable in the 1+ and the 3+ oxidation state. And you can also do permutations. So that gives you quite a lot more possibilities. So here we started to work. This was around 2016 or so. And so I asked my student Brenda to start looking for alternatives of what elements could we work on. So we settled on copper and antimony for several reasons. First, they're both abundant and relatively non-toxic. Copper and antimony have shown to be good absorbers for other PV devices. And there's also an abundance of copper and antimony perovskite, which suggested that we could potentially form a material like this. So Brenda went into the lab and started to try these elements, and she couldn't get anything going. She would only get separate phases or the same materials and so on and so forth. Until one day the copper one accidentally oxidized to copper 2 and then formed this beautiful polycrystalline black powder that she was later able to crystallize. Of course, this really caught our attention because if you want to do a solar cell, you want a material that absorbs a lot of light. And black is an indication that it's absorbing a lot of light, most of the visible spectrum for sure. So we were puzzled by these results and wanted to know what was going on with this. And after running single crystal X-ray diffraction, we realized that what we got in our hands was what we later called a layer double perovskite. And layer because it forms these inorganic layers separated by this space, and double because it has two different metals. Okay, so this was the first report of a layer double perovskite to the best of our knowledge, and we were really excited for this. I'm gonna give the detail of why this was the first case, but it was some sort of serendipity that is a particular family of perovskites in which, as you grow the thickness of the layer, you need fractional oxidation states, or as we later find out, you would need a combination of metals to access the thicker layers, which are usually more interesting for photovoltaic applications. So here's another way of looking at them in some sort of ways, like introducing vacancy and replacing them with different elements. And what this generates is this layer perovskites. And you generate layers by changing the charge on the metal. So this is something that is done in situ. You don't have to do much work. You pretty much just put the reagents there and you get your layer material. And what was really surprising is that the prime material, the one which has only antimony, has a really large bandgap of about 3 electron volts. But what we found is that our material, once we incorporated copper, it has a much smaller bandgap of about 1 electron volt. So this is a really drastic difference between these two materials. And of course, when you're talking about 1 electron volt, you around the region where making solar cells become something interesting and something definitely possible. We also show that the stability of this material was really great. It has a great thermal stability. If you want a material for solar cells, you want it to be stable to high temperatures because it's going to be heating while exposing to sun. And as compared to other perovskites, as I mentioned, so we expose our material to relatively high humidities, and we also combine humidity plus illumination, like 4 days of simulated sun or UV and humidity, and the material stay put in all of these conditions. We see no sign of degradation, which we think it's a really great sign of stability, and it's a really great step towards that stability that we're looking for in these materials. Unfortunately, not everything is roses, and as Uncle Ben said, with great stability comes great trouble. And that's what we have found with this material. The stability comes at the cost of a really diminished usability. So far it's been really challenging to make good films, to process the materials in a way that we can actually make decent solar cells. So this is definitely a challenge, and it's something that we are currently working on to solving that. But beyond just a particular application of that material, what we think these particular materials show is that there's a lot of new possible materials that we haven't really explored, that we haven't yet known. So I'm here presenting you what we call the double layer perovskite chemical space. So now, as I told you before, now we can start doing combinations, but now remember that now they're combining metals in a 2 and 3 plus oxidation state. So again, there's a lot of elements that are stable and that could potentially be of interest for these oxidation states. So I'm highlighting a few here. And the question then was, can we actually form materials with other elements, or is that the only member of the family that we just discovered? So Brenda was quickly to prove that you could also put manganese instead of copper, and when incorporating manganese, you also change the properties of these materials. Also, very importantly, you can modulate the amount of manganese. You can do solid solutions and progressively introduce different amounts of manganese on these materials. And what this results is a modulation of the bandgap of these materials and the properties of these materials. So you can see here that the manganese-only material has this pale pink color, the color traditional of manganese. And as you go to the copper-only material, you get the bandgap of 1 electron volt, and depending on the concentration, you get this plethora of different properties or different bandgaps. Of course, when you're talking about manganese and copper, you are also changing the magnetic behavior of this material, which is something that it's really interesting to explore. And what we did then was we wanted to understand these materials and understand their electronic structure. So we rely on DFT calculations, and we validate those calculations with extensive studies with XPS, X-ray absorption, and X-ray emission, which told us that we really had done a good job modeling these materials and that we really understand how to model these materials. And what this allowed us is to propose, once we understand this, to propose how we could possibly explore this space. So here's again this map. I now color coded it for different possible elements that would go in this material. But of course, as you can probably imagine, playing with all these permutations is not easy, and it can be a lot of work. So we wanted to find a smarter way to explore this space. So what Raul figured out is that we could do a combination of an experimental and a computational approach to reduce our space and try to see if we can find new materials. First, we did what we call an extended Goldschmidt study, in which, by considering the radii of the different elements, we can discern which ones are more likely to be stable. So pretty much we could see that the ones in the shaded or the gray area are the ones that should be stable according to these criteria. So now we're starting to reduce our search. Then she used DFT to calculate the relative stability of the materials as compared with the possible degradation compounds. And what ended up happening after all this is that out of 90 possible materials, we end up with 8 that our calculations and our estimation suggested that are stable. So 8 is a more decent number, and it's a number that we can experimentally try. And here's the list of materials that our calculation show or suggested that are stable and we should be able to synthesize. It was nice to see that these first ones that we previously synthesized that I already talked about are indeed predicted to be stable, which was it's an important thing, that our calculations were consistent with our previous results. And then what we were able to do is to show that all these other materials that we had in synthesis before showed to be, we were able to synthesize them and to prove that we can get to them experimentally, right? There's still a few that we've not been able to do. As you can see, well, there are chromium and iron, which their 2+ oxygen still is not their favorite. So there's definitely some work to do. But regardless, like using this approach, we were able to find 5 new different materials that were previously known and that form part of these new forms of materials that I've been telling you about, the layered double perovskite. So now you might think, and why should I care about these family of materials, right? Okay, well, I'm going to tell you a bit of a story about these 2 materials and why we think they could be useful. So these 2 materials are some of the 2 that I mentioned. They have a similar structure. They have manganese bismuth and cadmium bismuth. And when we combine them, we find that these materials can luminance very brightly. They show that they have photoluminescence of up to 80%, which is really high, and suggests that they could be used in devices such as LEDs or maybe as phosphors in LEDs or for other applications. And we did quite a bit of studies to understand these materials. We did quite a bit for the physics studies to understand these materials, and that we think that we now know how these materials behave at the electronic structure level, which I think is really important. But also we have shown that these materials are excitonic by nature, which is really exciting for a whole new different set of experiments, or a new different set of applications. So what I would like to tell you here is that I hope I've convinced you that layered double perovskites have great potential and there's plenty of chemical space yet to be explored. So here I show a diagram, and I didn't talk much about this. We have done only a tiny bit about this, where we have hybrid layered double perovskites. And here's a little review article that we wrote last year about it. But also, I hope I show you that the diversity of layered double perovskites make them attractive candidates for several applications. So I showed you already that they can be used as phosphors. But one other application that others have shown using the same materials is that they can be used for photodetectors. This is an application that has been quite successful. As I mentioned, solar cells is still a work in progress, but we're confident that we'll eventually get there. And other possible applications are in LEDs as photocatalysts and for applications in spintronics. So we're really excited about these new family of materials. And I hope I have convinced you that there's great potential and that there's a lot of chemical space that we have yet to tap and to explore. So it's not only about solar cells, but there's still way more things to be done. With that, I would like to thank first my research group. This is my research group, which has been amazing, especially during this pandemic. That has been really hard, but they have stayed put, and they're there on the Zoom meetings, which I really appreciate. All of my collaborators, staff, of course, funding from CONACYT and UNAM. And finally, here's some of my our contact details, our webpage, and our social media platforms. So with that, I thank you all. And I'm looking forward to hearing some of your questions or comments. Thank you very much.
Thank you, Diego, for this presentation. I think that there is a lot to learn about these perovskites, and I hope everybody can take the opportunity to ask some questions directly to Diego. If not, I could read them as in the previous talk. So Diego, I'm gonna jump in and ask you. So you and I, we have discussed a lot about, or sometimes about the characterization using different techniques, and of course, we have discussed the use of solid state NMR to characterize these. So sometimes it's possible. So how often do you think would you use it for the systems that you have in your lab?
I'm not using it nearly enough as I should. That's for sure. There's a lot of possibilities there, and I would certainly think that we should be using it way more. Most of these materials behave really well for NMR, especially, of course, for solid state NMR. So I think there's a lot of potential there, but to be honest, we haven't really tried much on it, so I may have to rely on you to explore that.
Yeah, no, well, I was thinking because you will probably have a different insight of the material. Maybe there is perhaps less deep than X-ray diffraction, but with the solid state NMR you can capture, I don't know, perhaps some impurities within the material or something like that. So, okay, we have some questions from the audience. So, from India, somebody asked you, does every structure following the ABX3 type is considered a perovskite? are there any exceptions?
Yeah, there are actually many exceptions. Like there are quite a few materials that have that formula, but that they don't have a perovskite structure. So having that formula does not guarantee that you have a perovskite. But there's some likelihood. Okay, so I don't have any. I don't have any material from the top of my head right now, but there's definitely quite a few that have that formula but are not a perovskite.
Good, thank you. So another question from Ana Sanabria. She says, excellent conference. Thank you. I want to know if you know there are natural types of these minerals.
Yeah, certainly there are natural type of this mineral. As I said, perovskite was first a mineral, calcium titanate, and there's also the barium titanate and even some lead oxides that are found in nature. So you can find them, and there's actually some places in the world there are tons and tons of cubic tons of perovskite materials. Not exactly the ones that we have as solar cell absorbers, but there's definitely quite a few in nature.
Thank you. So Gabriel Medina wants to ask a question.
Sí. Gracias, Braulio. So Diego, thank you very much for your webinar. So it's these systems are very interesting, but from the theoretical point of view, it's a little complicated to model them. So in your experience, what is the most serious problem in modeling this system? So because you need to consider many factors, no?
Yeah, let me go back a little bit. So if you consider, for example, these materials, which were the first that we tried to model, there are some challenges. So the first is that manganese and copper have magnetic properties. So before doing that, you have to figure out what is the best way to model the magnetic behavior. Are they paramagnetic, antiferromagnetic, ferromagnetic? And unfortunately, pretty much the only way to try that is just to run calculations, see how it behaves and what is the lowest energy. Of course, we have some clue from experiments, but even within, for example, the ferromagnetic properties, there is at least 3 different ferromagnetic properties that you have to, or arrangements that you have to consider. And for doing that, you have to construct a supercell of a few unit cells, which of course increases the calculation cost. So that's the first challenge. And then that only becomes more complicated when you combine these two metals, because now it's harder to understand the magnetic behavior, and I guess the structure possibilities increase, and therefore the magnetic possibilities increase even more. So now you have to try quite a few different electronic and magnetic structures. So that's a lot of calculations that have to be done, and to do that you have to grow relatively big supercell. Well, maybe not that big, but maybe something around 4 unit cells, or maybe 6, depending on what you're trying to do. And that definitely makes calculations harder, slower. So that's a great challenge. And then the next challenge is, here I have elements that are relatively light, but as we move on, we definitely would like to go to heavier elements such as bismuth, and definitely putting lanthanides in there would be something interesting. But right now it's really hard to calculate such elements because we just don't have enough calculation or computing capacity. So to summarize, I think those are the main challenges.
Okay, gracias, Diego.
Okay, I have another question from the YouTube audience. Professor Victor asks if you have tried to determine the quantum yield, the emission and quantum yield of these compounds in the solid state at different, of course, solid state at different temperatures.
Different temperatures, no. measuring PLQs at different temperature is not something that we're able to do. But if there's someone in the audience that can help us with that, we would love to. We've only been able to do that in solid state and at room temperature. In that scenario, the highest is around 80%. We would expect that increasing the temperature would increase that yield. But no, we've not been able to try that. That would be amazing though.
But that is because you have the technique limitations, or
yeah, we just don't have the technical capabilities, and as far as I know, it's not common to do that because you either you have a cryostat or you have an integrated sphere to do those measurements. And those two are hard to combine. I mean, I know that there's some people that can do it, but it's not trivial.
excellent. So I have another question from you from the audience in this in Zoom. Are all your structures solvent free? If not, is there any dependence on solvent content or solvent nature on the properties of these materials?
Yes, all of the structures that I told you about are solvent free, and actually most of the structures that we work on are solvent free. Usually you would want that because unless there's some really non-volatile solvent, having some solvent, as you said, could affect the properties, the structure, or even completely degrade your material when heating, right? So if you want to have a material that it's going to be exposed to sunlight for years, eventually, if you have a solvent, it would most likely degrade. So ideally, you don't want to have solvents in there. So far, they don't have solvents.
Okay, thank you. But there is a trend. I correct me if I'm wrong. There is a trend to introduce different types of organic counter ions, right? So to increase or regulate the optical properties of these compounds. And so what is I'm thinking about you mentioned methyl ammonium ions, but there must be something some others that are larger than those, or not? It's not possible.
We're retaining the same 3D structures. There's a handful of those. You can do from amidinium. You can, in some cases, do ethyl ammonium. There's some cases of diethyl ammonium. Vanadium, it's another case. But it's hard to really think about going to things that are larger than that, particularly for the 3D perovskites. Once you go to the 2D versions, there's certainly a lot more cations that you can incorporate, and there's something that we have been explored. I didn't talk about that, but there's definitely a lot of room to work on that.
But that is because you have problems for me, like you have segregation processes going on.
Yeah, so what happens pretty much is, let me go back a little bit. So if you consider the traditional perovskite structure, so in order to keep this inorganic frame that is shown by the blue octahedra, your cation must fit in this cavity that is defined by this cube formed by 4 octahedra. So if you change it for something that is really big, what happens is that you start, for example, layering, or even further, you can reduce the dimensionality further. So it changes the nature of the materials. We sometimes call those 2-dimensional perovskites, and I have a picture. They would look something like this. This is not a great depiction, but you would have inorganic 2-dimensional layers, and in the middle of the layers, you would have organic cations here. And here you can play a lot. You can put a lot of different things for sure.
Excellent. Okay, I think we don't have any more questions right now? I don't know if somebody else in the audience would like to ask a question to Diego. Well, if not, we thank you, Diego, for this such a fabulous presentation. It's quite a lot of work, and congratulations for being named Talented 12. It's very well deserved, and we are very happy for you because I don't think we haven't met another another such a young talent. So congratulations, Diego, for you.
Thank you, Braulio, and thank you to the entire organizing committee.
Thank you very much. Okay, so we have a few minutes before we start with the last session of a conference session today in around 15 minutes. So stay tuned, and we will see you guys in around just a couple minutes before noon. Thank you very much. Okay, welcome everybody again for the third and last series conference of this series of today's series and the Latinx Scam event on Twitter event 2021. Sorry, it's an enormous pleasure for me to introduce Miguel García Garibay, who has been very supportive for the Latinx Scam initiative, and he has very enthusiastic to give a presentation about his research at UCLA. So before we go to the slides, I'm gonna take opportunity to take the opportunity to present him. So Miguel did his undergraduate studies at the University of Michoacán, en la Universidad Michoacana de San Nicolás de Hidalgo, in 1982. He then continued his doctoral studies at the John Schaeffer's Group at the University of British Columbia in Canada, graduating in 1988. After that, he did a postdoctoral stay in joining Nicholas Turrow's group at Columbia University before joining UCLA in 1992. Naturally, his research has achieved international recognition, and he has been focused on the study of reactive intermediaries, solid-state organic chemistry and photochemistry, and crystalline molecular machines. He has received many recognitions and awards throughout his career. Some of them are the following. He was an associate editor of the Journal of American Chemical Society. Then a couple of years ago, he was named ACS Fellow. And last year, he was recognized as a member of the American Academy of Arts and Sciences. He holds currently the Dean of Physical Sciences at UCLA. And more than that, I consider him a friend and excellent advisor, supervisor, and a truly friend who has helped Mexican scientific community along these years. Thank you, Miguel, for being here. The floor is yours.
Thank you so much, Braulio. Can you hear me okay?
Yes.
Okay. I will share my slides now. Let's hope that everything is good. Go to presentation mode. How is that?
Yeah, that's perfect. Yeah, we can see it.
Oh, yeah, well, I want to start just by not only thanking specifically Braulio and Gabriel Merino for inviting me, but really thanking the large group of organizers from all of the Latinx community, both in Latin America and in the rest of the world. So I'm really proud to be part of this community. It's a real pleasure. I debated what language to use for this presentation, and you see I have a initial slide in Spanish, actually kind of Spanglish. I'm just noticing some errors there. But I thought that, you know, unless I could mix it with a little bit of Portuñol and others, probably it wouldn't work out as well. So I'm going to stick to English, if you don't mind. And this is the title I gave for my presentation. So my presentation today is intended to be sort of like a relatively didactic lecture where I'm going to be covering examples that are very old, some that are relatively new, and I might get to a couple of things that are unpublished. And what I like to do is to give you an idea, an outlook of solid state chemistry, and in particular solid state photochemistry. So my presentation plan is a little long. I might have to end a little early, but I think it's structured in a way that I think I will be able to deliver my message, which is really I feel working in crystals is extremely exciting. So, you know, when we think about crystals, the general practice, the general mindset is that crystals are really nice to have because you can put your molecule in a vial. It will stay stable for a long time. You can use it for crystal structure. So typically you want to get nice, chunky, big crystals that you can put in the diffractometer and get some beautiful structure that will adorn your manuscript and also give you some structural information. Today, I want to emphasize the fact that these crystals are really nice and beautiful, and etc. But if we use nanocrystals, these are crystals that are somewhere between 20 to 40 nanometers in the low end and maybe half a micron in the low end, so 500 nanometers. There is a lot of opportunities that make it possible to use solid state photochemistry under conditions that are just the same as the ones we would use in solution media, which makes it incredibly simple and friendly and easy to apply. So here we see another image another thing that, we like crystals because there is a lot of applications that one can use. We just saw the talk by Diego Solís on perovskites, and the fact that, the composition that you have in those crystals comes together to create some emergent properties is something really special. So this type of organization is fundamental. In my group, we're interested in crystals because we can plan control chemical reactivity. We can do green chemistry. We can take advantage of exciton diffusion to look at quantum chains. And we can look at mechanistic aspects by looking at precise structure reactivity relation. We have a program that I will not cover today on materials, crystalline materials, that we're looking at magnetism, multiferroics, molecular rotors, and molecular machines. So today we're going to focus on basically on the top 4 components. Now, if we think about it, the one thing that is probably most general and most common to most crystals is that they're characterized, especially molecular crystals, right? They're characterized by molecular order, molecular rigidity, and molecular homogeneity. That is, there is a very large reduction of entropy. If you imagine the total entropy of a liquid, right, when you have an innumerable number of configurations, conformations, collisional states, normal modes, etc., the entropy is just, the entropy content is very high. And when that liquid crystallizes, what happens is that everything collapses into a single structure, and the total number of states that are available in that structure are significantly reduced. So the first thing to recognize is that there is an enormous reduction of entropy. Well, let us think a little bit about more, right? So if we think about entropy, the first thing that comes to mind is thermodynamic entropy as defined by Clausius. So we would measure the heat content of our sample at a given temperature. And we have a number, it could be, I don't know, could be 150 calories per Kelvin. And when you do that, what you find is that it's possible that all of the crystals, all the beautiful crystals that I have in my image there, might actually have a very similar thermodynamic entropy. So the thermodynamic entropy is useful when we're looking for phase changes, changes of state, things like that. When we're looking for the entropy of activation, for example, that is useful, right, when we're making comparisons. But the entropy per se is not particularly useful if we use thermodynamic entropy. Now, what about statistical entropy as defined by Boltzmann, right? So this basically gives you the same number, but here the analysis is different. The PI in Boltzmann's equation represent the states of our system. And by states, we can look at the diagram on the bottom left. We're talking about the different vibrational, rotational, translation, and spin state that every molecule can occupy within a sample. So if we think about a perfect crystal at 0 Kelvin, all of the molecules are in the same state. So the fraction PI is 1, and the logarithm of 1 is 0. And that's what we know, that the entropy of a perfect crystal at 0 Kelvin is by definition 0. If we begin to introduce some heat, we begin to populate states. And now the PI, the fractions of those populations begin to get smaller and smaller. The entropy begins to get larger and larger. So at 0 Kelvin, we start with the dot in the center. Then we have a crystal, and our crystal begins to experience lattice vibrations and normal modes and others. So the entropy of the crystal begins to grow. The more heat, the more states are populated. Until then, we have enough heat to have a phase transition we go into the liquid phase. So the entropy of the liquid begins to grow as we put more heat, higher temperatures, and then it boils. And then we have the entropy of the gas that can increase also, right? So thermodynamic entropy tells us that the entropy of the gas is more than the entropy of the liquid, more than the entropy of the solid. And I said that's not very useful, because what is interesting to recognize is that actually the states that we have in the gas, in the liquid, and in the solid are actually not identical. So we're talking about different states of our system, right? So what that does for us is that, let me change here, is that we can change as we change the state of matter, we're also changing the number of states that are available. So we're changing the information content of our sample. And what I'm saying is that the amount of the statistical entropy of a crystal is smaller. The information content is also smaller, but it's more precise. We have a lot more precise information. It is interesting to recognize that the Boltzmann equation is identical to the Shannon equation that describes the information content of an information source. So it is intriguing to think about. So how do we go about thinking about information and entropy and statistical entropy in chemical system? Well, let me show you a really old example from my group. We're going to look at the photochemistry of carbons. So these are aryl alkyl carbons. As you can see, we have a diazo compound on the left. Photochemically, that goes in the carbon. The carbon can exist in a triplet and a singlet state. So 4 states actually, because triplet is 3 and 1 singlet. They have different reactivity, so that in solution it goes on to give at least 4 products, and if it, depending on the solvent, it gives many more products. So if we look at the top right, we are in the liquid phase. The configurational entropy is high. We have a high statistical entropy system that leads to low selectivity and low yields in solution. To analyze that a little closer, we can look at a Newman projection of our molecule along the bond between the diazo carbon and the alpha carbon, and that gives us something like that. So we will have a number of conformers that are under equilibrium, and they will be occupied depending on their energetics. Once we bring light, each of those conformers can correlate with a different carbon structure. The carbons may or may not equilibrate. That's going to be part of the entropy of our system. And each of those carbons can give rise to a different product, some too slow, so we are not observed. So this is when we have a system with a higher statistical entropy. So what's going to happen when we have a phase transition and we go from the nice big green circle to, say, a blue circle? We have a large reduction of entropy, right? So what the crystal has done for us is has reduced the statistical entropy, selected the system so that we can have 1 carbon, 1 diazo compound that goes to 1 carbon and 1 product. So what our beautiful ruby red crystal do for us is make our system much more tractable, more understandable, and really chemically more well-defined. So that when we do the reaction in the crystal, we can see that the crystal structure of the diazo compound correlates beautifully and uniquely with the crystal structure of the photo product in a least motion pathway. And we can do that in over 96% yield. So you can see here how a nice fine powder changes before and after reaction. And what you can see is that as a function of reaction progress, the selectivity of the reaction remains very high. We have excellent selectivity and high yields in the crystalline state. So the question is, well, so how do you go about, designing reactions in crystals? So what crystals are going to react and what crystals are not going to react? So this is the strategy that we use in my research group, and I invite you to test it. So the way we engineer reactions in crystals is by a very simple strategy. We identify substrates with intrinsic mechanistic pathways that upon excitation are able to break and make new bonds, even in a highly restricted environment, which is, by the way, what photobiology does. So photosynthesis, vision, all photobiology has schemes that look like this. So biology has developed this already very well. In our case, the information contained at the molecular structure level determines the reaction energetics. So what we see there, excitation and then going in a downhill stepwise process, is what the molecular structure is going to do for us. What the crystal structure is going to do for us, that information is going to determine the reaction trajectory. And that's what you will see in the examples that I will talk today. So this is a really good scheme to keep in mind if you want to play a little bit with reactions in crystals. Now, let me tell you about some interesting history that I think you will enjoy. So this gentleman, Leopold Ruciska, who is well known, is a fantastic chemist of the early 20th century. He won the Nobel Prize in 1939 for his work on terpene natural products. But he was critical. So, he was saying, well, chemistry only occurs in gases and liquids. And he is famous in my community, in the solid state photochemistry community, among other things, because he said, in German, of course, ein Crystal ist ein Friedhof, which means a crystal is a cemetery. So and that's probably true for many crystals, right? I showed you this picture before, and I guess you can see the analogy, right? So here we have a cemetery, and the tombs are nice, beautifully arranged, and you can say, well, yeah, all of our molecules are dead in the solid state, right? So they're dead. If you want to revive them, you have to put them in solution or in the gas phase, and then you can see reactivity. Well, is that true? Actually, that is just not true, and that's been known for a very long time. So the first history involves this plant, it's called Artemisia china, and these two gentlemen, Bartholomew Stromsdorf and Charles Pfizer, and sunlight is going to play a role. So we start by recognizing the fact that Artemisia is a plant that is distributed globally. And if you make a tea out of Artemisia, your tea will prevent or will cure worm infections, so helminthic infections. So it's a helminthicide, I suppose, is what would be called. But what Stromsdorf did is he took the plant, he extracted the plant, and he was able to isolate these beautiful crystals like this, right? So Stromsdorf isolated and studied these crystals. He happened to put his crystals on a little bottle and put them by the window. And what he discovered is that these crystals turned yellow and began to crumble and began to become a powdery. So something was going on. There was a photochemical reaction happening. And that's the first photochemical reaction ever reported. So a few years ago, we repeated the Stromsdorf experiment, in our lab, and this is how we set it up, right? So there is some xanthonin crystals in the microscope. We have a camera, we're taking pictures, UV light is coming on. What you will see is something like this. We will turn on the light, and this is what Stromsdorf saw. Look at these crystals, they're moving all over, the crystals are jumping in and out. These crystals are not only undergoing a photochemical reaction, they're transducing molecular energy into macroscopic energy, something that is called photosalience, a really interesting phenomenon. If we look at the reaction with single crystal X-ray diffraction, and here you can see how the molecules in the reacting xanthonin crystal transform into crystals of this photoxanthonin dimer. So we can say that, well, all that those crystals needed is some light to come back to life, right? So they were not so dead after all. So the next part of the story comes actually with Charles Pfizer, who's another German who immigrated to the United States, and he knew about xanthonin. He knew that it was used as for worm infections, and he knew that Bartholomew Stromsdorf had isolated. So he came up with the idea of mixing xanthonin with toffee to make some pills that could be given to the children instead of taking that very awful, bitter tea that they were given otherwise. And that's how the first pharmaceutical company was originated. So it turns out that xanthonin has some really interesting story. That was in the 1830s and 50s, right? So 40 years later, Giacomo Chamissian in Bologna did a large number of photochemical reactions, not only in solution, but also in the solid state. And he envisioned that the photochemistry of the future was going to be really important, that the sunlight was going to play a role. So it's interesting, right? So we have a chemical reaction and we have emerging phenomena. We can take molecular information into the macroscopic world, something that is called salience. And here is an example of the same xanthonin crystals. We're going to put light with a focus laser in a point and see what happens. When the reaction occurs, at some point, the energy that is stored internally in the crystal as a function of these structural changes can be transduced into ballistic mechanical information in those macroscopic crystals. So the fact is that actually our crystal comes alive. Well, not that crystal, right? We want to change crystals. These are the crystals that come alive. And there's a lot of beautiful examples of those, and I have a couple of examples shown here. The top example is a crystal reported by Professor Masahiro Irie in Japan quite some time ago. It's a photosalient reaction. And the one below is by our chairman, Professor Rodriguez Molina, who's looked at some beautiful examples of thermal salience, where thermal energy in the form of phase transition translate into macroscopic jumping and different types of motion. Let us get back to natural products and synthesis. So one of the major challenges in synthesis is the preparation of compounds with adjacent stereogenic quaternary carbons. And I'm showing a few of those examples here. There is a major challenge because the synthesis of these compounds require many steps. They're very poor stereochemical control. They're very difficult to purify because the reactions are so messy, the yields are low, which makes them to have a very low cost. So what is it about having two adjacent quaternary carbons that makes them difficult? Well, it's nothing but sterics, right? The fact that it's very hard to bring anything where something is already very congested. So the idea that we had in my group a long time ago is, what if we could take something cheap and simple like acetone and transform it into the type of adjacent quaternary centers that we see here? So that became a strategy of taking advantage of a photochemical reaction that is called a photodecarbonylation reaction. Here is an example of such a reaction. So this is an experiment carried out at UCLA in one of the gardens. That was in January. So that's the middle of the winter. After 2 hours exposure, this ketone with two alkenyl substituents, when we expose light, two bonds are broken, a biradical is formed, and that biradical carries the molecular information, and then the crystal makes sure that we get the stereoselective formation of the carbon-carbon bond, a relatively simple transformation. So this is really the message, right? One of my messages today is that molecular information determines reaction energetics and crystal information determines reactant trajectories. In this case, the trajectory is to preserve the stereochemical integrity of the reactant into the product. So here we have a cyclohexadienone, and we see the crystal, beautiful translucent crystal in the top left. We see the crystal structure with a van der Waals envelope. So our molecule has been selected to produce a natural product called cuparenone or alpha-cuparenone by taking advantage of this mechanistic diagram, this energetic diagram. So this is what this molecule contains within its own genes. It contains enough excitation energy to make an excited ketone. That excited ketone has an alpha bond that is so weak that is going to cleave to make this acyl alkyl biradical. Now that acyl alkyl biradical, the two electrons are looking at each other. So you think, well, it's just going to go back to the reactant. Actually, no, because this ketone is preparing the triplet state that cannot go back to the reactant. So instead, what it does is set up for a second bond to be cleaved to go to the biradical 2. That's what molecular information is doing for us. Now that we have the biradical, the crystal is going to keep the configuration of that radical to go on to the final product. Because if we were to do that in solution, the molecule will do its job, right? But the molecule is now in a very high entropy state in a liquid phase, and it's going to explore many different conformations and configurations, react with solvent and so on, and we're going to get completely different products. So in this case, our crystal is doing what an enzyme would do in a photochemical or photobiological reaction. We have no solvent, no metals, no reagents, quantitative yield, and no purification. So this is a wonderful reaction. However, not just as shown, right? If you see this particular, what you see in this image is that our beautiful translucent crystal becomes all opaque, loses its translucency. What that means is that light is not penetrating through very easily. So it would be difficult to scale up this reaction. If you think about it, it will be also very difficult to measure the kinetics of that reaction, to measure the quantum yields, or to do even any type of a spectroscopy. All of those challenges have their origin in things like the very high optical density and the optical properties of crystals, which includes dichroism, birefringence, and of course, a lot of scattering, right? So it's very difficult to introduce light into the crystal, and it's difficult to measure how much light is coming. However, all of these properties are size dependent, and they're a function of the ratio between the crystal size and the wavelength. That means that the smaller the crystal size, the smaller the problem is. One solution would be to just find powders, right? So we use fine powders, put them in the floor again, and we can do this reaction. It works pretty well, actually. So, we can do 100 milligrams in each of those glass plates. It's good, but it's just not enough, right? If we were to use, dry powders in a tumbling or flowing apparatus, it wouldn't work. They will tend to cake and eventually melt. So what we came up is with a different solution, which is nanocrystals. Prepare nanocrystals suspended in a non-dissolving fluid. That should help address these challenges. The strategy is very simple, right? So instead of having a large macroscopic crystal, we went to split that into trillions of nanocrystals and suspend them in water. In this scanning electron microscopic image, you can see that the crystals are on the order of 50 to 100 nanometers, and that's perfect. Under those conditions, our crystals in water look like milk because they're a colloidal suspension. It looks milk, of course, because white light is being scattered, right, of all colors, and that's what they look like. The way they are prepared is very simple. It was actually developed by Kasai and Nakanishi in Japan in 1992. The idea is to dissolve the sample, your molecule, in a small amount of a water-soluble solvent, for example, acetonitrile, THF, or ethanol, and add that into a solution. Add that solution into vigorously stirring or vorticing water. So when we have low loading, we have transmission of light, and we can do spectroscopy. When we have high loading, we can do photochemistry. We can determine the characteristics of our nanocrystals by using dynamic light scattering, atomic force microscopy, scanning electron microscopy, or we can centrifuge our crystals and collect them to get a powder X-ray diffraction pattern. Here is an example, actually, of a reaction that when we scale up. This was actually done by Lupita Hernandez, who's in the audience, and her husband, Gabriel. So they came to UCLA for a sabbatical, and Lupita and Gabriel decided to work on a large-scale photochemical process using a flow system. So using a photoenzymatic desymmetrization and simple chemistry, they prepared large amounts of this chiral ketone with 6 substituents in the alpha carbons. In the bottom, you can see the crystal structure of the reacting ketone and the photoproduct. The reacting ketone is chiral, the photoproduct is also chiral. And this is how they did it, right? So they devised a reactor that has basically the opportunity to pass the light through the suspension, through a reactor, and then through multiple passes have a really efficient reaction. Where when they collected this product, what they saw is that the test sample in solution in the top, as you can see, the NMR spectrum has literally dozens of products, meaning that it has dozens of different peaks from different products, a very messy reaction when you have a high entropy system, many configurations, many opportunities for reactions to take place. When you do it in a crystal, what you see in the bottom, you see the proton NMR of the crude sample, which is just perfect. If you just look at the methoxy signal in the middle of the spectrum here, you can see that corresponds to that carbon, and that tells us that the reaction in the solid state in the nanocrystals is just perfect, which is wonderful. Now, let me tell you that for these reactions to be so good, there is a couple of things that we need to consider. These are important. We need to mind the gap. So what's the gap? That's the gap between the liquid region and the solid region of the phase diagram. So if we have a pure crystal of a reactant and it's going to become a pure crystal of the product, we need to walk a phase diagram of a 2-component system. Something most of us learn in general chemistry, learn again in physical chemistry, and then many of us tend to forget. Well, Denise de Loera, who is from San Luis Potosí in Mexico and also came to UCLA, did a number of experiments to test some of these ideas. If you look at the phase diagram here, everything that is now covered in blue is that there is liquids there. Right? We need to avoid that. So let's say that you start with a solid. So you start a reaction at some temperature. indicated by that arrow. And you start on the light, and what you see is that you start in the solid, but then you go into a region where there is a liquid when you cross this line and so at some composition. So there is going to be melting, and then here there is going to be recrystallization. Let's take a look at that. Here we have an example of a fine powder. We're going to turn the light on, and we're going to see what happens. Our reaction begins to happen. We are not below the eutectic point, so we go into the liquid region of the phase diagram, and then the reaction C goes on, and then the product begins to recrystallize. But our reaction was very messy. This reaction was not very clean. In fact, this reaction lost the structural information of the crystal, and there were many reaction trajectories that could happen. So we need to avoid that. If you want a nice, clean photochemical reaction, you need to go below the eutectic point. I'll remind you that the eutectic point is the lowest temperature where you can see liquids and solids. So if you go below the eutectic, then you can be good. I have an example of that, but to show you that example, I'm going to take an opportunity to show you another really special reaction in the solid state. A reaction that we call a quantum chain reaction, which is can be viewed as a photon amplification system. We're using the same mechanistic information. We know that in this case, we have one or two intermediates, probably just one intermediate, but it is not able to go back to the starting ketone because once a bond is broken, it releases ring strain and it just cannot go back. So the diphenyl cyclopropanone that you see on the left, once excited, goes on to give acetylene or diphenyl acetylene plus carbon monoxide. What's cool about this reaction is that the quantum yield is 3.3, meaning that for every photon that is absorbed, there is 3.3 reactions. So that's a quantum chain. What makes this reaction special is that this reaction proceeds along the excited state. This is what we call an excited state adiabatic reaction. So we go to the S2 of the cyclopropanone, and in about 200 femtoseconds, it goes to the S2 of diphenyl acetylene. So diphenyl acetylene is the product and is still excited. After about 8 picoseconds, it will start to decay, but it has 8 picoseconds where it can do something interesting, which is transfer energy. So imagine a crystal where we have a number of diphenyl cyclopropanones. We excite one of them. Very quickly, within a few femtoseconds, we have an excited state. That excited state in about 200 femtoseconds is going to react to give an excited diphenyl acetylene. So we see now that the energy gap in the diphenyl acetylene in the blue is larger than the energy gap on the neighboring diphenyl cyclopropanone on black. So we will see a very fast energy transfer. At this point, we have one product and still one excited state. So what's going to happen here is that the quantum chain will proceed in a way that excitation will be transferred from one molecule to the next. And this will happen during the lifetime of excited state. So in this case, it's about 8 reactions per photon. All right. Now, these are some crystals of diphenyl cyclopropanone. This is a quantum chain reaction. So this is a really efficient reaction. And see what happens if we do this reaction well below the eutectic point, where we're showing here with the green arrow. Here it goes. This reaction is obviously completely destroying the reactant phase, and it's going into the product phase, into the diphenyl acetylene phase. But it's a solid to solid reaction. So that the time scale for recrystallization of the product must be very fast. But you can see how efficient solid state reactions can be. So there is really a lot of potential there. So when we look at the possibility of synthesis of natural products, our photoorganic synthesis, again, we're using the mechanistic scheme, right? The molecular information gives us the reaction energetics, and the crystal controls the trajectory. And here we see the synthesis of cuparinone, herbertinolite, and tochunyl acetate, 3 sesquiterpenes. All of them have adjacent stereogenic quaternary centers. And the key step of each of these reactions is a solid state photodecarbonylation. I will not go into much detail about that because you can find this in the literature. But I want to share with you something that is very recent. This we just published this year and last year. the synthesis of a very non-trivial, a very complex natural product called cyclotriiodine. So look at the complexity of this, this compound, right? It's, you see that there is an adjacent quaternary center, there is 4 heteroatoms, is chiral. Our retrosynthesis is based on one solid state reaction going back to this ketone. And you see the key reaction here in the crystal. So we prepare this dibromo ketone precursor photochemically, loses carbon monoxide and gives us this compound. And in a couple of steps, we get to this critical intermediate that is all set up so that spontaneously, basically, can go into the nearly into the natural product. So that can illustrate a little bit the sort of things that we can take advantage of by doing reactions in crystals. Here is a computational model where we can see how the trajectory of the bond recombination may lead to the desired product, although sometimes it might lead to something that is not desired. And if you want to read about that, you would have to look at the 2021 Jacks paper that was published just a few months ago. What about doing a spectroscopy? So what about learning some of the reaction mechanisms of these processes? Well, this slide just illustrates that we can measure the excited states. We can detect the excited states of molecules in crystals. So in this simple example, we have benzophenone nanocrystals suspended in water. And the beautiful blue glow that you see is the phosphorescence of benzophenone nanocrystals at room temperature. Here we see how by using nanocrystals, we can detect the phosphorescence in the bottom left. We can compare the phosphorescence of the nanocrystals with the phosphorescence in the bulk. On the top right, we can see that we can collect the UV spectrum by doing transmission spectroscopy. And if we do scanning electron microscopy, you can see that these crystals are about 200 nanometers, which is, again, smaller than the wavelength of light that we're using, that in this case is 355 nanometers. Because we can detect, we can do transmission spectroscopy. If we use femtosecond lasers, we can excite our molecule and detect the transition from the singlet excited state to the triplet excited state, which is shown here on the kinetic trace on the left. We go from the singlet to the triplet in a growth or intersystem crossing that takes about 10 picoseconds. If we use a nanosecond laser, then we begin to count when we're in the triplet state, and now we can measure the decay of the triplet back to the singlet. And as you can see, that happens in about 5 microseconds. And we can measure the spectrum of the triplet, so the triplet 1 to triplet N spectrum. So we can characterize the kinetics of this type of intermediates in the crystal state by taking advantage of these nanocrystals. What about the kinetics of these reactions that I've been talking about, right? These photodecarbonylation reactions. So this is what it looks like. This is what I illustrated, right? So we go from a ground state ketone to an excited state ketone that is poised to cleave a bond in the triplet state to give a triplet radical. We're calling radical pair. We're calling RP1. And that is poised to very quickly break the second bond to give RP2, what we call I1 and I2 on the top left, on the top right diagram. Putting some molecular information, we can have something like this. So let's say we start with this ketone that has phenyl groups on the alpha carbon. So these bonds here become really quick, very weak. So we excite our molecule and we have a very fast alpha cleavage, gives RP1. And then we have a very fast decarbonylation to get RP2. So in this case, we can very quickly get into the I2 intermediate or RP2, and we can measure the kinetics of intersystem crossing from RP2, from triplet RP2 to singlet RP2, which then goes on to give the product. And that's what happens, right? So in this case, we're using a neodymium Jack laser at 266 nanometers. We have a 10 nanosecond pulse, and we can see that the kinetics of this radical pair decay in a time scale of about 40 to 90 microseconds, depending on the nature of R. But there is a fast component that decays about 10 microseconds. We're still studying the origin of these double exponential kinetics. But the interesting fact is that these radical pairs have a very long lifetime, despite the fact that the two radicals are so close to each other in the crystalline state. So, these two radicals in the intermediate are no more than maybe 2.5 angstroms apart from each other. Now, what if we change the molecular information, right? And we make it difficult to decarbonylate. So we take a ketone where we select the R substituent shown in blue. We go to the excited ketone, right? So we call it here triplet 1. There is an alpha cleavage reaction that makes our first biradical. And then we make it unfavorable to lose carbon monoxide. So if you look at the top right diagram, I2 now is higher than I1. So what that means is that we've trapped I1, which is a triplet acyl alkyl biradical. And now what we can do is we can look at the kinetics of that biradical coming back to the starting ketone. In other words, we can look at the rate of intersystem crossing. How long is it going to take from one of those spins to flip from being the same spin to have one of them to the other spin? So we go from the triplet to the singlet, and then we can make a bond. We look at a number of ketones that you can see on the bottom, and we can get really quite interesting and valuable information on all of these. If you think about it, we're asking a lot, right? So what we're asking in this case is we're asking the molecule to contain the information to break a bond, even though it's in a closed packed crystal, so that when that bond breaks, those two electrons are going to be very, very close to each other. In fact, they're going to be basically a bond distance away from each other. This is as close as you can put two unpaired electrons, really, unless you put them, on the same molecular frame. But for a radical pair, you cannot go any closer than this. What are the consequences of having two electrons so close to each other? Well, what's going to happen is that if you think about this diagram, you look at the red line, that's the energetics of the triplet ketone as it begins to separate and is trying to break the bond. The black line represents the singlet state, right? When the bond is going to come back. However, when we break that bond in the crystal in our radical pair intermediate, it's in such a way that the energy of the singlet is far from the energy of the triplet. And the triplet sublevels are going to be spaced out because the dipolar interaction between each other is going to give rise to a large zero field splitting. What that means is that if you look at the bottom box, if the singlet triplet energy difference is too large, we cannot have one of the common mechanisms for intersystem crossing is hyperfine coupling, which is coupling the electron spin with the nuclear spin. That cannot happen. The energy is not appropriate. Now, the radical orbitals are collinear, and that's just not good for a spin-orbit coupling. For a spin-orbit coupling, we need that exchange of between two electrons produces some magnetic moment, some torque, and this is not happening if they are co-aligned. We have two different G factors that may help, and then the crystal rigidity will limit any possibility of spin lattice relaxation for those two spins to spontaneously relax thermally. So what we discover is something truly remarkable. What we discover is that, yes, we can look at these radicals. We can see that the spectrum is quite nice. And the decay is incredibly long. So up to 63 microseconds for two electrons that are staring at each other. This is truly unprecedented. So this is the longest lifetime for an acyl alkyl radical pair and biradical. What that means is that these electron spins are very strongly correlated, very strongly entangled. And that brings us interesting opportunities in the field of quantum computing. For quantum computing, we're going to need well-precise, well-defined quantum states. And the current type of quantum computing is based on semiconductor, basically on junctions. It can also be based on trapped ions. And there is a lot of efforts in using spins. The problem with quantum computing is that noise is really, really challenging, right? So the energy differences are so tiny that noise becomes a real problem. In this case, it's possible that noise might not be so important. Now, the Vincenzo's criteria includes scalability, which we think it could be possible in a macroscopic crystal. The qubits needs to be initially small, which we can do with a laser here. We need long decoherent time so that the different spin sublevels can have long lifetimes. We need quantum gates, and that could be accomplished through microwave and optical excitations, which could be both for the gates and for the reading. So the fact that we have the longest lifetime for acyl-alkyl radical pairs and biradicals, and that these two electron speeds are strongly correlated, make them actually pretty promising. So I'm gonna end there so that I've gone a little quick, but I'm gonna take a message from Diego's talk and leave enough time for talks. And I want to end really with this slide, right? Which is, it's not a summary of what I talked about, but is conceptually, I think, the most important concept, right? That crystalline solids give us probably the simplest way to control the macroscopic entropy of a system. That the molecular structure and the crystal structure combined contain chemical information that can be used to take advantage of for a number of possibilities, for chemical reactivity, for materials applications, perhaps for quantum computing and others. So I think this is a really interesting and exciting field. I've seen this field grow. I think there's a lot of very talented people working on it. And I hope that you will agree on that. Now, among the very talented people that have contributed to my group's research, we see a very nice picture here. We see actually Braulio here on the left, when he was a postdoc in my group. So we also hear Salvador Perez, also from Mexico. And there's been a really large number of postdocs, grad students, and visiting scientists, and colleagues in other institutions who have contributed to this work. So I'm very, very grateful to all of them for their contributions. And I'm very grateful to all of you for listening to my talk, and to the organizers for putting this incredible event together. So I'm gonna end there. I'm gonna stop sharing, and I will be happy to answer any questions that anybody might have. Thank you, Braulio.
Thank you very much, Miguel. It is time for there are a couple minutes for the audience to ask questions. I already have one question from the audience. Professor Maria Rosales Os from Simbestab asks, so what happens if you have a polymorphic forms during these reactions? Do they react similarly or not?
Well, first of all, hello, Mary. It's nice that you're part of the audience. So thank you for coming to my talk. That is a fantastic question. So different polymorphs, even though they may have the same molecule and the same molecular information, they may contain different trajectories. Right? So if a crystal is racemic, you get a racemic sample. If a crystal is homochiral, you get an antiomerically pure sample. Now, when you have bimolecular reactions, then polymorphism is really important, because there could be a polymorph that sets up the two reagents in the right distance and orientation, and another polymorph that doesn't. And that is the case in the very classic photochemistry of the cinnamic acids, which really are part of the history of the field. So you're absolutely right. They may react similarly, but if the trajectories imposed by the crystals can be different, then you can see different reactivities.
Okay, thank you. We have time for more questions from the audience. I'm gonna check because they're also asking from the YouTube channel, and sometimes I miss those questions. But yeah, well, maybe it's time for another question here. Yeah, well, Miguel, I really it's really amazing what you can do with a very quick instrument that measures all these transients. But those instruments are not. I mean, it's not. I don't recall the name of the instrument that you got at the like around 2013 that when you can measure these transient. So how do you measure it? I don't recall really.
In the organic chemistry community, is generally referred to as a laser flash photolysis apparatus.
Laser flash.
All right. In the physical chemistry community, they refer to it more commonly as a pump-prof system. So, in Mexico, we have some really very talented kineticists like the director of the institute, Jorge Peon, who use these techniques to look at that very fast kinetics.
But in that case, in that scenario, you will have to work with a suspension with an emulsion, like you mentioned it, or you could work with crystals. You
can work with crystals if you make the crystal sufficiently thin, right? So there are techniques that you can make a crystal that is half a micron thick, but it is very challenging because you have all the parasitic optical challenges coming from dichroism, birefringence. The results depend on the orientation of the crystal because the crystal is anisotropic, right? So nanocrystals suspended in water make it possible to look at solid state kinetics and photochemistry the way you would look at proteins, right? So there are protein complexes that are 40, 50, 100 nanometers, and you do everything that you do in solution, you suspend them in solution. So the nanocrystals are a little bit like that. The nanocrystals can be thought of as a state of matter in transition between supramolecular systems and bulk solids. So they're very friendly. So any laser flash photolysis system can be used to measure transient kinetics in nanocrystalline suspensions. Thank you.
Thank you. There's another question also from Professor Mari. Thank you very much for your excellent talk. She says, have you been able to follow these crystal-to-crystal reactions by using synchrotron X-ray radiation?
Actually, we really haven't. So there are groups, in various parts of the world that have been able to use a laser pulse and then use synchrotron pulse excitation and collect data on the kinetics from the crystal structure. So we have not been able to do that, but it is a wonderful technique that is getting more and more powerful. It's currently challenging, right? But it's nonetheless very interesting. I can comment quickly that what we have been able to do is we can get the crystal structure of crystals that are 100 to 200 nanometers by using microelectron diffraction. Right. And I have to brag that one of the inventors of microelectron diffraction is my colleague at UCLA, Jose Rodriguez. Jose is, of course, Mexican who emigrated to the United States when he was a child, and he's just a brilliant, brilliant structural chemist.
But in those cases, I recall that by dealing with very, very small crystals, you could probably have a different polymorph than the one that do grow at large specimens for single crystal X-ray diffraction in-house, right?
That is possible, yes. So, one always need to confirm whether the identity of the phase in the nanocrystals is the same as the identity of the phase of the bulk solid or large crystals. Okay, thank you. There is another question. So from Professor Gabriel Merino, he says, thanks, Miguel, for a very interesting talk. The examples you show are such as that you can remove CO. Is it possible to remove other stable molecules like water, or is only what is special? What is so special about CO? Actually, there is nothing special about CO. It's just that it's a model system that we've been looking for a long time. But I showed an example of a diazo compound where you extrude nitrogen. We look at diacerines that also extrude nitrogen. There are examples where you can extrude carbon dioxide. So it really depends again on the energetics of the molecule and whether the bond dissociation energies, and there is a state correlation between the excited state and the intermediates where the bonds are cleaved. But no, there is really nothing special. So you can think about losing SO2, SO, sulfur dioxide, sulfur monoxide, things like that. So a small, in fact, you can think about also losing ethylene or acetylene in a retro Diels-Alder type reaction or things like that. So, the fact that from what we can tell, what's important is that the molecular energetics are such that the excited state correlates with intermediates that are downhill, and there is no significant activation energy along the excited state surface.
But and also for the stability of the radical that you end up with, right? So if in the cases you show, you have a phenyl groups and methyl group. So it's a tertiary radical or secondary radical, I don't recall, but
right.
That can also contribute to the stability.
Absolutely. So it is interesting because the stability of the radical is what determines the exothermicity of the reaction, right? So our observation and calculations also indicates that you really need in a crystal pi substituents that will delocalize the radical by resonance, because a tertiary radical will not cleave. But if you have a vinyl, alkynyl, aromatic, or cathodidative effects, then they will cleave.
Thank you. There is another question from Professor Jorge Tiburcio Investap. For the photosalient effect, do you have an idea how to harness the mechanical force to obtain macroscopic and directional motion?
Hi, Jorge. Thanks for the question. That is a great question. In fact, the crystal that I show in one of the slides from the work of Masahiro Irie, in the same paper where he published that example, he had a beautiful example of how he used a crystal to activate or to actuate a gear. So photochemically at a distance, he was making the crystal basically bend in a way that it would then push the gear step by step. So he was basically transducing photochemical motion into macroscopic mechanical motion. And the way he was doing it again is by using light coming into different directions of the crystal to control the bending angle and the direction of bending. So there is a lot of interesting possibilities.
Okay, I think there are no more questions from the audience. Thanks again, Miguel, for such a wonderful talk. I think there's a lot to love about crystals and to learn from them. And we thank you for the support on this Latinx Chem event, and hope you can continue supporting these types of events. Thank you very much.
Absolutely. Thank you so much. Thanks, everybody.
Bye.
Bye-bye.